guys? Hope you're all doing fine. And we're back again with another video. I just have to mention one tiny detail. Well, the last time you watched the video, I think it was probably yesterday for you, but actually it was 14th of August 2017. And today is 21st of September 2022. Time travel, son. And before we go ahead and continue with the videos, I just have to mention another thing, and that is I've heard from many of my friends and some of you guys that one of the problems you have with these lectures is the speed of these videos. Well, I'm sorry. I know that I'm not the fastest talker ever, but you can always go ahead and just, put the playback speed on 1.5. I myself listen to these videos on 1.5. So the next topic is cell cycle, and we are going to go through the cell cycle the same way that the department intended going through it. So we're going to discuss the basics of cell cycle. We're going to discuss then the mechanisms, the more detailed mechanisms of cell cycle, and at the end we're going to discuss the regulatory mechanisms that control the cell cycle itself. And as you guys should already know, the best way to go through the material is to go through the material together with the minimals. So I'm going to actually give you guys the definition of cell cycle from the minimals and go through it. So cell cycle is the life of a eukaryotic cell, so it's the life of our cells, which lasts from the formation of the cell, which means the end of the previous cell cycle, right? So there's a cell that is formed now, it means the previous cell cycle is over, to the next cell division. So whenever this cell also finishes the division and makes daughter cells, then that would be a full cell cycle. It can be divided into two phases, the cell cycle, right? It can have a resting phase, which is the beginning phase, which is also known as the interphase, by the way. The interphase is when the cell is resting, is going through G1, S, G2, and then it has an actual cell division phase. The cell division phase is also referred to as the M phase. Now, M phase can also be subdivided into other phases, which we will go through as we are going through the lectures. The resting period can be further divided into three phases, as we mentioned, the G1, S, and the G2 phase. Now, I know some of the smarter ones listening are thinking to themselves, well, isn't there a difference between the mitotic division and the meiotic divisions when it comes to the cell division categorization? And the answer is basically, when we're talking about the fundamentals, no. So the fundamentals stay the same, and actually here you can see the life of the human cells as a whole. So we start as gametes, and fertilization happens, the zygote and the embryo in the womb, and in the early stages of embryogenesis, the human cells, the embryonic cells start developing this other cell line. So try to think of it as two main cell lines: the somatic cell lines and the germline cell lines. So the somatic cells are responsible for your body functions, right? Your somatic cells are the cells of your heart, the cells of your GI tract, your epithelial cells, and your germline cells are different, even in the number of chromosomes, and we'll talk about it. And you probably already know the basis of this from the genetics, but we'll go through some revision. So the germline cells have to go through meiosis, and what they're responsible for is to produce the offspring, or produce the cell offspring that is going to form the gametes, which going to repeat the cycle in order to maintain this, if you try to think of it as a epigenetic phenomena, this cycle is trying to maintain the cellular genetic integrity of the species. But I don't think you really need to know this for the exam. I'm just putting it forth in order so that you understand the material better. So let's then talk about the most basic form of these types of divisions, the mitotic and meiotic division. So this slide you're seeing here, this is a mitotic division. As you can see, in mitotic division we have chromosomes, and we have chromosomes in all sorts of divisions, but basically the chromosomes will replicate, and the homologous chromosomes will be separated during the M phase in a way that from each cell, let's say you have 10 chromosomes, you'll get into 20 chromosomes. So basically, whenever you're having a mitotic division, you're getting one cell replicating its entire material into two daughter cells. And how exactly is that different from the meiotic division, you may ask? Well, it's because in the meiotic division, actually, by definition, the number of chromosomes is reduced to the half. The number of the chromosomes of the progeny cells, of the daughter cells, is actually half of the parent cell. And how it works is that it actually goes through two consecutive cell divisions. During the meiotic division, the cell goes through two consecutive cell divisions. Whereas in the first one, as you see these are the chromosomes. In the S phase, the DNA, replicates and stuff. So we have these homologous chromosomes. As you can see, these arrows are pointing at each one of these, two chromatids together, which form the chromosomes. And these, the blue one and the red one, are homologous chromosomes. Now, since the DNA has replicated, each chromosome it now consists of two chromatids. As you can see, the blue one is like two rods, and the red one is also two rods. So these rods, each respective rod to its partner, is actually referred to as a sister chromatid, as I'm pretty sure you already knew that. So the first cell division, what happens is that the homologous chromosomes get separated, right? It's going to go through some, many levels of, prophase, prometaphase, anaphase, but we're going through the basic concept right now. So after this, during the second cell division, now the sister chromatids separate and form the final progeny, and these are the gametes. And actually, in case of sperm and egg, it's different because as you probably know, not all the gametes from the egg, from the oocyte, is viable. One of them becomes viable, but you'll actually read about it more in embryology. And this other slide is basically, explaining further what we just explained. So this is mitosis, this is meiosis, and in meiosis, the tetrad means, which means the four of the homologous chromosomes, each containing a pair of sister chromatids, will eventually separate to, each sister chromatid being a progeny or a daughter cell itself. But in mitosis, as you can see, you have a chromosome, the chromosome replicates, and the replicated chromosomes, get separated into two progeny cells or daughter cells. So now that we have covered the most fundamentals of what meiosis and mitosis is, let's go through again the overall of what these steps are, and we're going to break down these steps even further. So basically, you start with a cell, right? And I'm going to add some context to this picture over here. So the cell starts here. And I'm going to assume that G1, G2, S phase, at least the basics of it. So this cell is right now where? At which stage? Give yourself 2 seconds. Think about which stage of cell cycle this cell is. Because here it says chromosome replication. So at the point of this arrow, the chromosome replicates. So this cell is in G1. And in G1, there's a checkpoint. And we'll talk about checkpoints later. In G1, there's a checkpoint in which if the nutritional status or if the environmental status around the cell is good enough for replication, that checkpoint would be elevated so that the cell could proceed to the next levels of cell cycle. So as you can see, when it says chromosome replication, it means it's going through S phase. And then after S phase, here the cell enters into G2. And at the end of G2 is also a checkpoint that determines if the cell can go through the next level, right? And what is the next level? The next level altogether is called M phase. And by the way, the first level, the G1, S, G2 stages, they were all together called the interphase. Now we enter M phase. What happens in M phase is two main things happen in M phase. Well, three main things. The mitosis itself, which is the division of the nucleus. So we have a nuclear division. The other thing that happens is cytokinesis, which is the division of cytoplasm. And the other thing that happens is the division is actually the replication of organelles and some other stuff that we have to talk about, actually, about the microtubules, that is not in this graph. And by the way, the point about karyokinesis, I've seen that this term has been used interchangeably with mitosis, but I personally don't think these terms are can be used interchangeably. But since I think they have been used interchangeably in your minimals, you can go ahead and use them interchangeably. So here you can see another of these graphs. This actually is designed for you to understand or grasp a basic concept of how long does each stage take. I really seriously don't think you need to memorize in hours, because there's another graph that shows how many hours each stage take. But I think what you should know is that actually the longest stage, and I think this is a bit contorted. So the longest stage is S, the DNA synthesis stage, which makes sense because you know the DNA is long and it needs to be synthesized. So it's something around 7 hours. G1 is something around 5, and G2 is something about 3 hours. And what's interesting is that the entire mitosis itself, the nuclear division and everything, it happens in around 1 hour. So I think your takeaway from this and the next slide that I'm going to skip should be that the DNA synthesis stage is the longest stage. It takes 7 hours. That's it basically. Now, as you can see, we're going into more and more detail as we're going through the slides. So here, what do we see? We see the same stuff we talked about. We see G1, we see S, we see G2, and then this is the M phase, right? And there are some good explanations here. So G1 is growth, and by the way, I think you should also know the actual medical term you can use for this is called hypertrophy, when the organelles or the cell itself gets big. So G1 is hypertrophy. The S phase is synthesis of DNA and histones, and what it says, the propagation of epigenetic state, duplication of centriole. So basically right now you don't need to focus on the propagation of epigenetic states, because it's gonna have to do with the epigenetics, and I think it's more important for your genomic studies, not for your cell bio. But what's important, very important for S, is this part probably you already knew, synthesis of DNA and histones. But this part is also important, duplication of centrioles. And the entire centrosome complex, which we'll talk about later, will start getting activated during the S phase. And then there's the explanation for G2, which is cell size control. And what it means is that basically the cell has to always maintain, its growth. So there is a chance that the cell grows too fast and it explodes, or something, right? So these mechanisms are put in place so that after each stage, the cell stops and sees if it's, the situation is good enough, the neighborhood is, chill enough, so it can go ahead and go through the other stages. And as you can read here, the moving of centrioles to the poles also start, which we again are going to cover later. However, this slide introduces a new concept, G0. So what the hell is G0? So we can get into a lot of detail about G0, but think of it this way. Not all cells are going to divide in our body. So many of our cells, for example, all of our CNS neurons are going to always maintain a non-dividing status, right? So they're not going to divide, meaning that they're always going to be before the S phase, meaning they're going to be in G1 or G0. Now, the important things that you have to know about G0 is that there are some stuff, but the two most important things is that the content of the total RNA of the cell in G0 is the least compared to all stages, even to G1. So this is actually a way that you can figure out if a cell is in G0 or not. The other thing that is important about G0 is the existence of the primary cilium. And we'll talk about the primary cilium and like what they are exactly. But for those who already know a bit about centrosomes and centrioles, well, you should think of it this way. When the cell is not dividing, it doesn't need the centrosome complex to exist. So it kind of, stores the centrosome complex in a form called the primary cilium. And now we get to this beauty. Well, I don't think you need to read any of this. Don't quote me on that. I'm just saying it. But I think this slide is basically put over here in order to explain your minimal 202, which talks about the start or commitment or restriction point of the cell cycle. And it's very simple. It basically says that after we pass after the cell passes the G1 checkpoint. Whenever the cell sees the situation is good enough so that it can go through G1, it will go through S anyways. So there are no like limiting factors for cell going through the S phase if it has already passed the G1. Even if you add some, substances or substrates that are cytotoxic to the cell, that are toxic to the cell doesn't understand that the environment is toxic, so it should stop, replicating the DNA. It will commit, and then it will commit until G2, and in G2 the cell will, stop again and see if the situation is good. And because we added the cytotoxic material, the situation is not good, so the cell will not divide. That's basically for you to understand the commitment points. These checkpoints are the points in which the cell gets committed to go through the next phase. And again, in this slide, you can read that most of the differentiated non-dividing cells of the adult body possess primary cilia. Why is that? Because most of these cells are non-dividing, so they don't need the centrosome complex, so they possess primary cilia. So now let's talk a bit about the S phase and some very obvious stuff about it. For example, this slide that says transcription of most genes does not vary greatly through G1, S, G2 phases. Oh, really? So the transcription doesn't vary even in S phase? Exception. Histone mRNA synthesis is increased in S phase. Holy cow, good thing that they mentioned it. But basically, yeah, this is the dot position, and I don't know why they even added this graph for it. So although this is just an info, slide, I'd like to talk about it like for a second, because it says mitochondrial division is coupled with cell division and it's controlled by connections to the cell cortex and the ER, which is, and endoreticular, the ER, that. But the thing is, it is not supposed to be like this, because the mitochondria, it's not supposed to be exactly part of, or at least it's not supposed to be integrated as part of the human cell DNA. It has its own DNA, it has its own semi-independent functions, but through some connections to the other organelles or other regulatory mechanisms to the cell, even the mitochondria itself is actually coordinated to replicate, to duplicate whenever the cell is duplicating. Otherwise, our cells would end up with many uh, mitochondria, because they would be decoupled from the cell division. And now we're going to discuss a bunch of slides that are about, the methods, the scientific techniques or technology we can use in order to observe the cell division, and how much DNA and how much histone exists in each stage of the cell division, so we could understand exactly when each stage ends and when each stage begins. And we can also go with some research on the cells regarding what happens if you inhibit some specific CDKs, which we'll talk about CDKs a bit later. So my advice for you guys is that accept flow cytometry, which is actually an important thing, and you're going to need it for, I think you needed it for chemistry, you will need it for microbiology, you will need it for internal medicine, you will need it for everything. But except this flow cytometry, the other things are not important. You just have to memorize like 3H thymidine or like microscopic techniques, which is like it has the name on it, as methods in order to study cell division. So for example, this is an example of a study done with the 3H thymidine, and I think one of the main things you can get away from it is the duration of each phase based on the study, which is like the S phase has the highest duration. Now this is a flow cytometry chart, and you can see here that the chart that is over here is the same thing, but basically the area under these curves, so the more area is under each respective curve, it means that the higher the duration of that stage was. Again, making the case that the S phase has the highest duration, around 7 hours. So this is actually something we already discussed. How to detect cells in G0. So here it says in rapidly dividing cells, the RNA making up ribosomes accounts for almost 85% of the cell's overall RNA pool, which means the opposite is also correct, which means in the cells that are not rapidly dividing, the RNA content is very low, which is something we discussed. In cells with G0, the total RNA content is way lower than the cells in other stages. And these are some of the ways through which you can achieve cell cycle synchronization in order to study the cell cycle. For example, we can deprive the cells from the basic material they need in order to, go through the cell cycle so they would stay in G0. You can add some cytostatic or cytotoxic material or substances to the cells. Cytostatic means that the material, the substance that we add to the cells is in a way so that the growth stops. It's a static, situation with the cell. And cytotoxic, well, it's toxic to the cells. Some of these drugs are like these ones. You definitely don't need to remember, like nocodazole, for example. But you should know that if you wanna, inhibit the progression of a cell to the next phase, you can use some drugs in M phase or in G2 phase or in S phase in order to prohibit that progression. Some other techniques is literal separation of, for example, the detachment of the rounding up mitotic cells. So you have to have very technical, microstructural, laboratory material in order to be able to do that. And they actually have some laboratory material capable of doing that in the Department of Physiology and also in the Department of Cell Biology here in Debrecen. This is, for example, one of the studies in which they added some FBS. FBS is like a growth factor cocktail. It has many growth factors in it. And when the control cells were grown in a medium with 10% FBS, the DNA content was more. Whereas the cells which were, cultivated in a free medium, they had a less DNA content. It's kind of intuitive. And well, if they ask you what kind of cells we do experiments on in order to figure out, how the cell cycle works, you can say we work on, human cells, we can work on mammalian cells, we can work on yeasts, and we don't just work on, normal physiological cells. For example, we can work on mutated form of yeast cells. We can work on different, specific types of mammalian cells, which is what this slide is here to talk about. And here, actually, in case of yeast, you can cause some mutations by just changing the temperature, and in that way, you can actually study exactly how mutations work in the cell cycle. These again are some examples of how we can alter the cell cycle and study it. So, for example, in vitro, so it means in the lab, they added some progesterone to the cells arrested at G2 stage, and when they added the progesterone, the cells, continued and they grew. And they, for example, in another assay, they added MPF, which I think is maturation promoting factor. It's another growth factor, and adding this growth factor also caused the growth and progression to the next stages of cell cycle, and they studied it. This again is an example of what we talked about with, for example, the yeast. Use of conditional mutants to study cell cycles. E.g., I think they meant temperature. Yeah. So, for example, just by changing the temperature, which means that you're changing the condition, you can cause mutation in some cells, for example, yeast cells, and that way you can study, the cell cycle. So here are some other duh statements here. G1 cells are competent for the initiation of DNA replication. We've covered this. You have to pass through that G1 checkpoint in order to go through, but if you can pass through that G1 checkpoint, you're home free for DNA replication. G2 cells, which means they've already, gone through the S phase, must transit through M phase. So they must first go do mitosis and karyokinesis and everything before they can go to G1 and S phase again. Duh. So that would be the end of just the basics of cell cycle.